Hi, I'm Allison Mundy, and Jordan Webb and I are going to be presenting over the research we did at Lyon College this past summer. Our research was over the recent expansion into eastern Arkansas of the poultry houses. Currently, Arkansas is the second leading producer of poultry in the nation. And so we tested over a gradient of these poultry house densities, different streams, to see how our stream houses are doing. These triangles right here represent the streams that we tested, and all the red dots are representative of the poultry houses in the area. And here are some pictures thanks to Katie Cruz. So the first part of the research was we were looking at invertebrate communities. Now, looking at invertebrate communities can help paint a picture of what the streams are like because we have bugs that are pollution intolerant and pollution tolerant, meaning that some bugs do very well despite there being a lot of pollution in the water, and some bugs don't. So if we take a sample of bugs in an area, we can count to see how many of those bugs are pollution intolerant versus pollution tolerant. We do this by using what's called EPA taxa, which means we count caddisflies, mayflies, and stoneflies. You can see we have a picture of a stonefly right here. Another thing we look at is abundance and diversity. So the reason we look at these things is it tells us abundance is how many bugs are in the stream, and diversity, which is how many different kinds of bugs are in the stream. So what we found was that what we have is on this graph we have poultry houses, the streams from poultry house low density to high density, with dials right here being our control group. So we look at our control, we can see it has lots of different colors, which is good. We have lots of different bugs, high diversity. And as you also see, we have a very large EPA taxa, or those pollution intolerant bugs, which means the stream is very healthy. So what we expected was this EPA taxa to go down as we have higher densities, and for the colors to kind of homogenize as we get across too. And that's not necessarily what we see, because I want to point out the Tennessee, Hubble, and Upshaw. These have very, very low EPA taxa, and they have very homogenized colors. This one only has 3, we have 4 here, and 3 here. I want you to remember these streams because they're going to be important later in the water chemistry data. And as we continue to look, we have the abundance and diversity. There was no significant relationship between poultry house density and the abundance or the diversity of macroinvertebrates. But we have Hubble and Tennessee up here, which are way above where all the other ones are, meaning they have high abundance. And over here, they are lower than kind of everyone else, are all the other streams. So they have low diversity. So we have high abundance and low diversity. That means those streams have homogenized, meaning there's a lot of one kind of bug, which isn't great. So that leads us to the water chemistry part. So what do we look at for water chemistry? We looked at nutrients, which is phosphorus and nitrogen. We looked at suspended solids, which is how muddy the water is. We looked at the algal biomass. We looked at pH and dissolved oxygen. In this presentation, I'm only going to be going over the nutrients, which are the phosphorus and nitrates. And so this is what we found. We found that looking at phosphorus, that Hubble and Tennessee had crazy high amounts compared to all the other ones. So this kind of leads us to believe maybe Hubble and Tennessee are outliers, like they were in the bug data. So we took out Hubble as an outlier, and this is what we got. Even with Tennessee in here, we do have a significant p-value. So with that, it leads us to, does poultry houses affect the health of our streams? And we think they do. We know they do from the research done in Oklahoma with Tyson. But what does that mean here? Well, the bigger question is, why is Tennessee and Hubble such outliers in our data? We think it's because it doesn't have to do with density, it has to do with proximity to the streams, which means it doesn't have to do with how many poultry houses are in an area, but where the poultry house is in relation to that stream. Meaning that we should, with the help of Dr. Davis and Zachary Ward, next summer, pick better streams to sample based on this information. So that way we can paint a better picture of how exactly these poultry houses are affecting our environment. And with that, we will go see Jordan Webb on crayfish.
Hello, I'm Jordan Webb, and I'm going to be talking about the crayfish side of our project. Now let me share my screen with you so you can see my PowerPoint presentation. Now before I talk about our PowerPoint, or our project specifically, I want to talk about the paper that really sparked our interest in this project. Now this paper is by Dr. Lignot, and he was looking at how different pollutants were putting stress on different crustacean species. So more specifically on how it affected their osmoregulatory abilities. Now what is osmoregulation? Osmoregulation is the organism's ability to filter ions in and out of its environment. Now I just picked 3 pollutants from his paper that pertain more to our study, which is potassium, ammonia, and nitrite. Now he used 2 different crustacean, lobster and shrimp, and he used various levels of the pollutant. But as you can see with potassium, there was no effect on its osmoregulation capacity. But with ammonia and nitrite, it did affect their osmoregulatory ability. In fact, it decreased their osmoregulatory ability. So we got to thinking, with all these poultry houses popping up, having this nitrogen and phosphorus runoff, could this be affecting the crayfish species living in the area? So what we wanted to do is find a crayfish species that was native in all 12 creeks. Now that crayfish species turned out to be the Ozark crayfish. Now the Ozark crayfish can be light red to dark brown, but its most distinguishing factor is the dark speckles on its claws that you can see right here, and the 2 black bands on its back. Now these crayfish can live up to 2.5 years. They can grow from 1 to 3.5 inches, and they're omnivores, meaning they eat both plants and animals by mainly little insects in the water, and they are bioindicators. This is the most important thing. And you're probably thinking, what is a bioindicator? Well, bioindicators are really cool because this organism, its health correlates to the health of the environment. If the organism is thriving and doing well, then the environment must be healthy. If it's struggling to survive, dying at a very young age, then their environment must be harmful. So this is how we decided to approach this project. First of all, I want to point out the 12 creeks that or 12 streams that we looked at for our project, and they're represented by green triangles. Now the little red dots are poultry houses, and as you can see, there's several poultry houses around this area. And so before we even caught a crayfish, we looked using ascorbic acid and cadmium reduction method. We found the levels of phosphorus and nitrogen in the water. And using that data, we picked the 3 creeks with the highest, lowest, and middle levels of pollution, because we wanted to see if those broad ranges, if osmoregulation is really affecting the crayfish's ability to osmoregulate. Now, to determine if it's affecting their ability to osmoregulate, we're looking at the NKA ATPase, the sodium potassium ATPase, which is the motor protein in osmoregulation. So if they have a high abundance of this protein, they would be very good osmoregulators. So what we did is we got 3 crayfish from each of the 3 creeks. We extracted their osmoregulatory organs, the gills, antennal glands, and intestines. We put these organs in a paraplast block. We put that block in the microtome that you can see here, and it would cut itty bitty little slivers about a cell thick, and then we would put these slivers on a slide, and then we would stain the slide using histology staining and immunofluorescent staining. Now histological staining would just be normally staining the tissues, but immunofluorescent staining is really cool because we can actually tag the NKA protein and actually see the abundance on the tissue sample, which is very cool. Now before I go into our pictures of our slides, I want to show y'all where the gills actually are located on a crayfish. Now this crayfish isn't the Ozark crayfish. I got this from a study that Kansas State did, but it would be located at the very same spot in the Ozark crayfish. But you can see the gills are located right here. Now the gills are very feathery and fluffy, all fluffed out. They look just like a feather. And here you will see our normal histological stain. The daffy in blue, this is tagging the nuclei in each cell, so it's stained all of the cells. And then the NKA we tagged in green. So when we overlap the pictures, you can see which gill lamellae have NKA and which don't. And you can see that the NKA is located on the outer part of the lamellae, in the outer basal domain, specifically in the ionocyte. And as you can see here, some of the lamellae don't have NKA at all, which is interesting to see that some may be used for respiration, as you see here, and as the ones with NKA would be used for osmoregulation. Now this picture up on the top is a zoom-in view, and the NKA is in red in this instance, and you can clearly see that it's localized in the outer basal. Next is the green gland or intestinal gland. This functions very much like our kidneys. So you can see right here in this picture of a crayfish that the intestinal glands are in the very front behind the eyes under the brain. They're very, very small. There'll be 2 of them, and they're 2 itty-bitty white ovals. And as you'll see here, this is their normal tissue stain, the DAPI tagging the nuclei in blue, NKA here in green, and on the overlay you can see that the NKA is localized in the labyrinth. The labyrinth is this maze here, which is very interesting because the function of the intestinal gland is filtering through the crayfish's hemolymph, which is their blood. And their blood filters through here, and it goes through this maze, you know, back and forth, and it would make sense for the NKA to be located here to absorb all the ions out of the hemolymph before it is excreted from its body as dilute urine. So the next or the final organ we looked at is the intestine, and you can see the intestine is located in the crayfish here. It's this little tube, and here is the normal tissue stain. Here's the DAPI tagging the nuclei in blue and the NKA here in green. And on the overlay, you can see the NKA here is in the interior part of the intestines. And as you can see, it's specifically in the enterocytes. Now, enterocytes are absorptive cells, so they're absorbing the ions. So it makes sense that the NKA was located. So now what did these results mean? Well, comparing all 3 streams, we found no differences, major differences in the level of NKA or NKA found in these organs, which means that there isn't a big difference in their ability to osmoregulate that we can tell. So this has us thinking that maybe the pollution impacts on a transcriptional level, as seen in fish, or might impact other physiological mechanisms such as acid-base regulation or respiration, because these poultry houses, they have phosphorus and nitrogen runoff, which creates harmful algae blooms. And these algae blooms are harmful because it's depleting the water of oxygen. So it's stealing the oxygen out of the water, which could be a big problem when the crayfish are trying to respirate and pull the oxygen out of the water. So what does the future look like for us? Well, we will continue studying this summer, even on into 2021. Allison will be evaluating relationships between invertebrates and their habitat, and we will be analyzing the levels of mRNA and protein expression for NKA and other osmoregulatory proteins in gills, intestines, and green glands to further evaluate the impact of water quality on osmoregulation. And so I would just like to take the time to thank the United States Geographical Survey, the USGS, and the Arkansas Water Resource Center for funding our experiment. They have been so kind to give us everything they need. And I would like to thank y'all so much for listening, and I hope y'all are staying safe and washing your hands. Bye, guys.